Assalamu alaikum dear students. Today we will discuss about DNA vectors. DNA vectors are similar to the regular vectors. Vectors are anything which can carry. They are simply the vehicles. They don't get themselves affected from the things that they are carrying, but they can carry it from one to another. For example, mosquito is the vector for plasmodium. Plasmodium causes malaria. So the mosquito acts as a vector by carrying plasmodium from an affected person to a non affected or healthy person. It is also the carrier for dengue fever virus of the vectors or the DNA of interest or the gene of interest, whatever you want to call it. When we say gene of interest, it is abbreviated as GOI. So, a cloning vector makes multiple copies of a DNA, whereas expression vectors are the ones used for making RNA or protein out of it. You are well aware of the term expression. By DNA expression, we mean transcription and translation. Transcription is the process of making RNA. Translation is the process of making proteins out of RNA. So these both of these processes, transcription and translation, are the way DNA expresses itself. So when we use expression vector, this means that we want either RNA or protein products made from this vector. In this particular type of vector, you can see um, in fact, I would like to phrase it like there should be three important things in a vector. If any three of this is missing, we cannot call that DNA as a vector for genetic engineering. First of all, the vector should have origin of replication. Second, it should have multiple cloning site. Third, it should also have some selectable marker. For majority of the prokaryotic vectors, we use antibiotic resistance gene as the selectable marker. So we can easily differentiate cloning vector and expression vector. As I have told you, one is used for making multiple copies of the DNA itself, whereas the other one is used for making proteins or RNA out of it. So the types may include plasmid, cosmid, phages, BACS, YEX, or MAX for cloning vector. Remember, BACS are bacterial artificial chromosome, YEX are yeast artificial chromosome, and MAX are mammalian artificial chromosome. Whereas the expression vectors um, are the ones which has the proper setup for a DNA to be expressed. And by proper setup, I mean that there should be a promoter, there should be transcription terminator, there should be other regulatory elements which are needed for the expression of a DNA. For example, initiation sites, transcription initiation site, translation initiation sites, enhancers. There should also be ribosome binding sites to ensure proper translation. And we can also add other things which could help in the downstream processing of that protein. For example, specialized tags or the secretory elements. This timeline indicates the history of vectors. The first vector, which was used in genetic engineering, um, dated back to 1973, and that first edited plasmid was PSC 101. Then in 1977, PBR 322 were surfaced and they were followed by the PUC series. Then in 1989 PRS series came, and that was followed by the phosmids and the modular vectors. And with the rise of synthetic biology in 2000, there then we can see the tailored or custom made vectors, for example bioprex, PCC1 phos, PSEVA vectors, etc. And now we have the minimal modular vectors. So the which vector to be used, that depends on the what is the experimental design which we are working on, and how we want to clone, how we want to screen the gene which we are cloning, and what is the ultimate goal, whether we want a protein or we want a multiple We want multiple copies of the DNA for later usage, etc. We can also call the vectors as of prokaryotic origin or eukaryotic origin, or and that also depends on where we want to use our vector in, like what will be our host cell. If the host cell is going to be prokaryote, then we will use specialized prokaryotic vectors. And if we want our proteins or our vector to be used in eukaryotic cells, then we have to use specialized eukaryotic vectors so that they can work inside that host cell. And we can use these vectors for permitting combination of cloning, DNA sequencing, in vitro mutagenesis, as well as transcription and episomal replication. Episomal replication means um, plasmid replication. Fine. Episome is anything other than the chromosomal DNA. In past, the plasmids were also called as episomes. So we have discussed that three essential properties of vectors are required. One is its autonomous replication, which we call as origin of replication. Um, the presence of origin of replication, ORIC, on the vector ensures that it will replicate autonomously, like it won't depend on the cell, the host cell, to replicate, to ensure its replication. It can initiate the replication of its own by itself. Then the multiple cloning site or the polyclonal sites are the restriction sites, which are present on the vector, which we can utilize for cutting open the vector, and then we can utilize the same restriction enzyme if its cutting sites are present on the flanking regions of our gene of interest to cut the DNA, and as a result we can easily join that fragment with our vector. The selective markers are important for picking out whether the cell has taken up our vector. This table indicates some commonly used vectors, for example, plasmids, lambda phage, cosmid, P1 phage, BAC, and YACS. And you can see that the amount of DNA they can carry varies, with the smallest amount of DNA uh, was carried by plasmid, whereas YACS can carry up to 1000 kb. Here in detail too when we discuss the blue white screening, so I'm not I'm relying of that on that description. Kindly go back to those that slides and lecture to refresh your concept. Gene of interest, we have discussed that